Bismillahirrahmanirrahim. Assalamu alaikum, dear cadets. Now we will start chapter number 3, that is enzymes. Before moving into the definition of enzymes, we will define catalyst. What is a catalyst? A substance that speeds up a chemical reaction without any change in it is known as catalyst. Now, what are enzymes? Enzymes are biological catalysts that speed up the rate of biochemical reactions without being permanently changed in the process. Enzymes are biological catalysts because they are protein molecules made by living cells. Because they are protein molecules made by living cells. Enzymes are very important because in their absence, reactions in the cells would be too slow to sustain life, that is, making life almost impossible. Now, what is the meaning of term enzyme? The term enzyme was first coined by Kohne in 1878. It is a combination of two Greek words. En means in or inside. An enzyme means yeast. Yeast is a unicellular fungus. In 1897, two brothers, Buchner brothers, that is Edward Buchner and Hans Buchner, added yeast extract or juice. Yeast is a unicellular fungus into a grape juice, and the yeast extract caused the grape juice fermentation and formed alcohol. So it was concluded that yeast extract or juice contains some active ingredient which caused the fermentation of grape juice. It was given the name zymase and later on enzyme. So that's why the term enzyme means inside, enzyme means yeast. The study of enzymes is known as enzymology. Now, characteristics of enzymes. Enzymes are proteins in nature except ribozymes, which are RNA. Enzymes function as catalyst and increase the rate of chemical reactions that would otherwise occur very slowly. Number third, enzymes are globular in shape. Globular means globe-like, and enzymes are encoded by DNA. As you know, enzymes are proteins, and their informations and the information for protein synthesis are encoded in DNA molecules. Enzymes are highly specific in action, that is, each enzyme will catalyze only one type or one kind of reaction. For example, protease will catalyze only the breakdown of proteins, not of carbohydrates, lipids, etc. Next, enzymes are required in small quantity, that is they are very efficient. Enzymes are affected by the temperature, pH, etc. Enzymes reduce or lower the activation energy required for a biochemical reaction to take place. And last, enzyme molecules have a specific site known as active site. These sites have specific shapes. It recognizes and binds substrate molecule and then carries out reaction. Now, structure of enzyme. As you know, enzymes are proteins, and proteins are polypeptide chains made up of amino acids. What is active site? Active site is also known as substrate site or catalytic site. Active site is a specific site where the substrate molecule binds, broken down and converted into product. Active site is a specific site or region in the enzyme molecule where the substrate molecule binds, broken down and converted into product. Or in other words, the region of the enzyme where the substrate binds to its specific enzyme is known as active site, and it is this active site which has a specific shape. Now, this is enzyme molecule. And this region of the enzyme molecule, and this is substrate molecule. This is substrate these are two substrates, substrate A and substrate B. So this region of the enzyme where the substrate molecule will bind is known as active site, and each active site has 3 to 12 amino acids. So each active site has a specific shape. There are two substrates, substrate A and substrate B. So the shape of substrate A fits into the active site of the enzyme. So substrate A binds to the active site of the enzyme by hydrogen bonds, making an enzyme substrate complex, and later on it will be converted into product. As the substrate B cannot fit into this active site, so it cannot bind into this enzyme active site. So therefore, as the active site have specific shape, therefore enzymes are highly specific in function. That is, each enzyme will catalyze only one type of reaction. Protease, for example, only for proteins, lipase for lipids, etc. Now what is substrate molecule? The molecule at which enzyme acts and transforms it into product is called substrate. For example, protease break the protein into amino acids, so protease is enzyme while its substrate is protein. Cellulase is an enzyme, its substrate is cellulose. Maltase is an enzyme, its substrate is maltose. Similarly, lipase is an enzyme and its substrate is lipids. Thank you.